So welcome everybody to today's webinar. Today is World Cancer Day. World Cancer Day, and to mark the day where IHMRI is very proud to deliver this webinar. The World Cancer Day is held on the 4th of February every single year. And it's a global initiative led by the Union for International Cancer Control. And its goal is to raise worldwide awareness, improving education and access to life-saving cancer treatment, and make sure that care is equitable for all, no matter who you are or where you live. And that's one of the main reasons that we're all here today. So my name is Kate. I am the Community Engagement and Events Coordinator here at IHMRI. I'd like to acknowledge the Dharawal people, the traditional custodians of the land on which IHMRI sits. I would also like to pay my respects to elders past, present and emerging, and extend that respect to any Aboriginal people present on the webinar today. So cancer is one of the leading causes of death worldwide. We have all been impacted by this devastating disease in one way or another. And this webinar will cover the impact that cancer can have on a community, but we're also going to strip it back a little bit and talk about what cancer actually is, what treatments currently available, and why there's a need for more research. And we're also going to show an example of how our IHMRI researchers are working together with clinicians and the community to improve treatment and outcomes for patients. So on the webinar today, we have Dr. Anne-Catherine Piper on the webinar today. We have Jeff and Dave representing the Illawarra Cancer Carers. I'm sure many of our audience members are familiar with the Illawarra Cancer Carers and what they do. They provide incredible support, all trained volunteers, providing support to people of our area, in addition to supporting research, and we'll definitely get into that in a little while. I'm just going to start us off with a little poll. And if you're comfortable to answer this, we would really like to know a little bit about you, our audience today. So hopefully this should pop up on your screen now. We're just really curious about why you're here today. So I'll just give you a few minutes to answer this poll. 1, you have cancer yourself. 2, I'm caring for someone with cancer. I know someone with cancer. I'm interested in research, or just for general information. So I'll just give you a few more seconds to answer that. Okay. All right, so we'll end that and I'll share the results. So quite varied. And overwhelmingly, the main reason that people are here today is because they're interested in research. So I'm really excited when we do get to talk with Dr. Anne-Catherine, who's going to share the research that she's currently working on and the importance of that research. So if you do have any questions about research or anything else that's covered today, please feel free to use the Q&A function. Just if you hover on the screen, a little toolbar will come up along the bottom. If you click on the Q&A function, you'll be able to post any questions. Feel free to do that at any time in the webinar. We'll keep an eye on it and we will answer your questions either as we go or at the end. And you'll also see the chat function. Oh, there we go, just open that. Hi, Dennis has just said a big hello to everybody. Please feel free to use that chat as a platform to share any experiences you have or make any comments. We really want to hear from you today. We want this webinar to be as interactive as possible, so please feel free to ask those questions and share your experiences. Hopefully that all sounds okay. So I think, yeah, my notes have completely disappeared. Anne-Catherine, I wonder, could you just tell us a little bit about who you are and the work that you do?
Okay, thank you, Kate, and thank you everyone for joining us today. I've never given a webinar before, so I just imagine the audience nodding and smiling along. My name is Anne-Catherine Piper, and I'm working at IHMRI on Wollongong campus. And my supervisor is Associate Professor Danielle Brunks, who's a clinical oncologist at the Wollongong Hospital, as well as in the Shoalhaven region in Nara, and a brilliant researcher as well. Together we work at the cancer research team under Senior Professor Marie Ranson, who's also a wonderful cancer researcher. And I've been lucky enough to work here as a postdoctoral fellow. So that means after I completed my PhD, I got a position here 3 days a week, because I also have by now 3 little kids at home. So it makes for a good work-life balance. And today I would like to talk to you a little bit about my research. But first, Kate had asked me, I had once given a talk at the University of Third Age and given really like a introductory seminar about what we're doing and what cancer actually is, what the treatment options are. And she asked me to sort of go back to that a little bit and really, yeah, as she said before, strip it back, get to the core and talk in plain English what this terrible disease is and what we're going to do about it. So I think I can get straight into it. Yeah, Kate, is that the idea? Yeah. All right, to start with the first question. So what is cancer? Cancer is actually a group of diseases. There's over 277 different types of cancer. And they all have one thing in common, and they're all characterized by the loss of normal cell growth control. So in a normal cell, it divides when it has to, so only when required. It undergoes programmed cell death when it's either damaged irreversibly or it has reached the end of its life. So if there's a finite number of cell divisions that have already taken place, our normal, well-behaving cells will die, but not so the cancer cells. So they do not respond to our normal control mechanisms within the cell, and they just keep on dividing and growing in an uncontrolled fashion, causing tumors to grow and the cancer to spread in the end. There's cancer can be defined as a genetic disease. So the genes is what makes us. So it's the recipe book that encodes for everything, all the proteins in our body. And 5 to 10% of cancers are caused by inherited mutations. So that means you get a mutation is like a spelling mistake in a book. And this is having in our genes, and that can cause either the complete loss of a protein or a production of a faulty protein. And so 5 to 10% of cancers are inherited, which means these are like the childhood cancers mostly. And the rest of them are caused by acquired mutations. So throughout our lifetime, we're exposed to UV radiation or cigarette smoke or alcohol. These are things called carcinogens. So they can cause something that can cause cancer. They cause mutations in our genes that encode for proteins that control the mechanisms of how cells divide. And if they are faulty, then the cells can turn cancerous and grow and grow and grow. I guess who's affected is probably more a question of who's not affected. Like everyone knows someone who has cancer. Unfortunately, in my family, there were several cases, but friends of mine, young, it's everywhere. It's everywhere. So in 2020 alone, there were 150,000 new cancer diagnoses in Australia alone. And mind you, Australia only has a population size of roughly over 25 million. So that accounts for 0.6% of all of Australians who were diagnosed in 2020 with cancer. So it's just ridiculous. And then, I did a bit of Googling, and the 50,000 deaths in 2020, that's the equivalent, caused by cancer. And that's the equivalent of 2 overfull wind stadiums, the one down at the beach. 2 of them completely chock-a-block full, and they in 2020 alone died because of cancer. But it's not all doom and gloom. Like, we're getting better at treating it, finding it, and destroying it. So we're trying to find new ways, improve treatment, improve drug treatment, trying to find it as early as possible, because that's our absolute best bet in finding a way to get rid of it. And since the early 80s, actually, the overall death rate has fallen by over 24%. So that is a number of deaths per 100,000 people. So that's fantastic. And we're going to improve this in the future it's going to get better and better. Absolutely. So today sorry?
So I was just wondering, Anne-Catherine, with that statistic, I mean, that's wonderful to hear. Is that from a combination of earlier diagnoses or improved treatments, or?
Yeah, so I reckon it's a combination of exactly that. Like you get better imaging system, you pick up smaller tumors earlier, there's more awareness. I think spreading the information and being aware of that you might want to go and see your doctor, or that your mum had breast cancer, so you're more likely to maybe have it too, or get it in the future. So I think there's a combination of that, exactly. More awareness, better picking it up, better imaging, better treatment. Definitely the treatments have improved so magnificently, like the drug development. So that's definitely a big deal there as well. So yeah, back in the 80s, the survival rate was less than 50% if you were diagnosed with any old cancer, whereas now in some cancers the survival rate is higher than 90%. So that's great. And 7 out of 10 Australians will survive for at least 5 years. So that it's getting, it's not all doom and gloom, even though it sometimes really feels like it, and particularly if we know so many people that have it or had gone through it. And yeah, touching base a little bit on the treatment options that are available. And I'm just going to talk about the big 4 first, and go a little bit into the ones, into the less known therapies, but I guess that would probably go beyond the scope of this webinar. So what I'm going to talk about next is surgery, chemotherapy, radiotherapy, and a little bit about immunotherapy. But then the lesser known kinds of cancer therapy include targeted therapy, hormone therapy, precision medicine. Sorry, is a question of stem cell transfusion or biomarker testing as well for cancer therapy. And how do they work? Well, I guess surgery is pretty self-explanatory. You have a lump, you want to cut it out, as much of it as you possibly can. But that's sometimes tricky to do, particularly if that tumor is too big, or it's close to vital structures, or very infiltrated by the blood vessels. So sometimes you need to shrink it down first. And that's often done with chemotherapy, which is a systemic drug treatment. So systemic means it affects your whole body, and you either get it via an IV drip in your arm, or you can also take oral tablets. And these treatments, so there are several types of different chemotherapies, but mainly what they're trying to do is trying to stop your cell from dividing. And often that is done by alkylating agents. So they add an extra base onto your DNA and stop the DNA from being able to divide. And DNA needs to divide in order for the cell to divide and replicate. So it stops the basic process of how the cell can divide. So, and that sometimes it stops the mechanism that stitches the DNA back together afterwards. So that everything around this process of DNA and cell division, that's where chemotherapy comes in. The problem is, though, chemotherapy stops all fast-growing cells from dividing. So that means that causes side effects for your body. So your hair will fall out because the hair follicles are fast-dividing cells. Your mouth might get ulcerated because all the mucus lining cells in your mouth are fast-dividing. Or you get really low blood counts because the cells in your bone marrow that produce the blood cells are very fast-dividing. So sometimes you might get the news, oh, he couldn't go for his next treatment because his bloods were too low. That's because the chemo hits those cells as well as the cancer cells. So the cancer might stop growing, but you get all these nasty side effects. So that can be one problem with chemotherapy. But obviously, they're developing better and better drugs that have less side effects and different, combination treatments can be done too. So often chemotherapy is given in combination with radiation therapy, and that is when you see Martin Carolyn, an absolute genius physicist, the chief physicist at the hospital in Wollongong, and also in the Shoalhaven region. And he and his team, they deliver radiation treatment to patients. So what that means is you get photon beams mostly that go in and destroy DNA or cause DNA damage in your cells, and that is done in a very targeted way. So unlike chemotherapy, which is systemic, radiation therapy is very targeted. So they do imaging on you first, so CT scanning or other types of scanning, so they can see where your cancer exactly is. They make a map and calculate exactly precisely to like the fraction of a millimeter where exactly your tumor is, and only there they will apply the radio beam, never nowhere else. And they have systems in place on how to protect you from any of the other radiation. And that in combination with chemotherapy is a very powerful tool, because so you're causing additional DNA damage, which will eventually cause cell death, because the DNA can't it's beyond repair eventually, and then the cell will die. And that in combination with the chemotherapy, which has already caused the cell to have DNA, trouble with the DNA replicating or trouble repairing, in combination, that's really powerful and gives you the chance to maybe lower the dose of the chemotherapy and lower the side effects that might come with it. So often radiation therapy is used in combination and after surgery. And the next step, I want to talk to you about immunotherapy, which is relatively new and a very, very promising field in cancer, and quite complicated when it goes down to the nitty-gritty. But today I'm going to just talk to you about in general terms. So immunotherapy is a biological therapy, and it's pretty much aiming at training your own defense mechanism, so your own immune system to go and kill your own cancer, which is a bit tricky because our immune system is trained to not attack anything that is your own body cells. But you can do it in two ways. You can either increase the production of T cells that will kill cells, and you can teach. So there's two different ways. So you can teach your immune cells to recognize your cancer by taking them out of your body and changing them in the lab, genetically modifying them to go and find that particular cancer that is just yours, and they go in and kill it. Or you can use checkpoint inhibitors, which will pretty much turn the off switch off. So your immune system has a way of checking whether or not something is foreign or yours. And cancer cells constantly tell the immune system, that's okay, I'm all yours, don't worry about me. But you can turn that checkpoint off. They're called checkpoint inhibitors, and then the cancer cells become targets again. So that is in use already in Australia and in clinical trial, and has shown very promising results. So I hope, that was a bit wishy-washy explanation, but I'm not an expert on immunotherapy yet. Because my project is using a different type of chemotherapy for a very nasty kind of cancer, which is what I'm going to talk to you about next, unless there's questions for me for the different.
Yeah, we've got a couple of questions here. Thanks, Linda. We've got a couple of questions from Linda. And one of them is around the choice between oral medication or IV to receive, I imagine, to receive the chemotherapy or chemotherapy medication. She said, does it depend on the type of cancer, or can patients generally have a choice? Is that something that you're able to answer, Anne-Catherine?
Yeah, I think I can answer it broadly, because actually from the University of Third Age, that was also one of the questions. And so it does depend a little bit on the properties of the drug, like the chemical properties, whether or not it can be given as a tablet or it has to be given as a liquid. And then I actually learned about that too, and he just said as well, like, you have to be able to 100% trust that the patient takes the drug and only at the right amount, and not think, one pill kills a little bit of cancer, maybe 5 pills kill a lot of cancer, so you really have to, it's good to be able to really control, because you're really poisoning yourself in a way. You're trying to kill the cells in your own body, so you really have to only give as much as needed and don't take more. So there is certain drugs can be taken orally, but you really have to make sure that it's only taken in the right, responsible way. Yeah, I hope that answers the question as much as possible. And yes, some drugs just cannot be administered in pill form, unfortunately.
Thank you for that answer.
No problem.
And Linda's other question is around the prevention of cancers. And Linda, forgive me, you might need to rewrite the question, but she says, is there any research-driven advice to prevent cancers? So I guess, is there any advice that has been brought together from evidence-based research around the prevention of cancer?
Oh, well, that's a great question. I think prevention is always better than trying to cure it once it's done. So very simple things, obviously, like don't smoke, don't drink too much alcohol, don't eat too much cured meat. And I'm in Germany, so I used to smoke, and when I came to Australia, I stopped because it was so expensive. and before I started doing cancer research, and I think, oh my goodness, I hope it's not too late for me. But yeah, so there's a couple of very obvious choices. Don't use sunscreen, because UV is an actual carcinogen. It will cause mutations in your skin. Use sun protection, that sort of stuff. And a general healthy lifestyle, healthy diet, high fiber diet has been shown to reduce your risk of any gut-related cancers. So that's sort of more obvious, I guess, lifestyle choices. But then there's also things that we can't control, like the genetics of it. Some people are more predisposed to, might have certain changes, or are most susceptible to get cancer from smoking, and other people don't. So there's more to the story, I think. And I'm personally very interested in why some people just never get cancer. So we always talk about these very grim statistics, but there's a large amount of people that just don't ever get cancer. So what makes them so special? So I think in my wildest dreams one day I would love to find that out, because there have been studies showing that the immune system in a certain type of rodent is just more responsive. So that type of rodent doesn't get a certain type of cancer that the other rodents get. And that is due to genetic changes, good changes in genes that encode for immune cells and immune response. So that is something I would be interested in one day, finding out, like, why don't some people get cancer? Why don't we all end up getting it?
Everyone knows that person in their life who smoked all their lives and drunk alcohol, and then lived to a ripe old age of 99.
That's right. That's what my mother always says. Like, see, he smoked all his life. So she keeps on smoking, even though I tell her better not to.
Yeah. So I guess it's all about just reducing your overall risk factor when you put them together.
Yeah, the things that are not, like, try to prevent the obvious risk factors. I guess
there are things that are in our control, and some things that aren't, and some things we just don't know anything about.
That's right, exactly.
Yeah, which is why the research is just so important. So
that's right.
And Katrina, thank you so much for giving us that overview of what cancer is and the treatment that's currently available. But the research that you're doing focuses on a particular type of cancer that there currently isn't any treatment for, or very limited treatment, and the outcomes are not so great. Can you tell us a little bit about that research?
Yeah, thank you very much, Kate. Yes, this is my new baby, so to say, my new project. And Emory generously has funded the project money for this. It's called a clinical translational grant, which I have together with clinical professor Martin Carolyn that I talked to you about earlier. And this was Dan's idea, because Dan is the man on the ground seeing patients every day, and he knows where there's obvious holes to fill, where's new treatments most urgently needed. And glioblastoma multiforme is the most aggressive brain cancer in adults that you can have. The 5-year survival rate is less than 5%. It's terribly grim. The median survival rate is less than 15 months from diagnosis on. So it's rough. Like, it's a really shit one to get. The limited treatments options is another thing. It's really hard to even develop new treatments options, because it's brain cancer, and you don't want to poke and prod around in the brain if you don't absolutely have to. So it's not easy to develop new treatment options. And at the moment, there's one gold standard treatment, which is a chemotherapy regime invented by someone called Stupp. And it's his protocol that it's used in the clinic using temozolomide. It's an alkylating agent chemotherapy, which alkylating agents are the most common form of chemotherapy. They're binding something to the DNA so it can't divide anymore. And that is in combination, also like debulking surgery is first, then you get radiation and temozolomide in combination, and then followed by another lot of temozolomide. But unfortunately, despite treatment, the disease most of the time comes back. And there's absolutely such high demand of finding other treatment options, new treatment options. And the fastest way to do this is using a chemotherapy that is already approved. And that is what Dan has suggested, and what we're now going to try is a combination chemotherapy. So that means that it's more than one drug in a combination drug. So this drug is called FOLFIRI, and it has 3 ingredients, and 2 of them are also chemotherapy drugs that cause DNA damage and DNA loss of division, being able to divide, and another drug which makes one of the drugs more powerful. So it's like a triple threat drug, and it's already approved and working very well for colorectal cancer and gastric cancer. So this is what makes it such a powerful alliance, because I wouldn't have necessarily picked this one or known about this drug, but Dan is the man on the ground administering these drugs, and I'm the researcher in the lab coming up with new ways on testing these drugs in the lab. So that's what we're going to do now. And we've purchased 12 different cell lines, and these cell lines are derived directly from patients' tumors. So they're called primary cell lines.
Can you, sorry to interrupt, Ankarin, can you just explain, what does that mean? You've bought some cell lines.
Yeah, so
how does that happen? Where do you get them from?
Yeah, so often, so there's things like a cell bank where you can purchase kind of standard cell lines that you use in the lab to test certain things. So you can buy kidney-like cell lines, skin-like or skin cell lines. So these are immortalized cells that will divide forever. They're not cancer, like normal cell lines are not necessarily cancer cell lines, but they have been immortalized in other ways that are not making them cancerous, so that you can use them as guinea pigs in the lab instead of using real guinea pigs or mice often. So that's often the pre-step before you go into animal work. You do cell culture work, and for that you use kind of standardized cell lines. And in this particular case, we are very lucky because the Berghofer Institute in Queensland, they have derived and established these cell lines and have characterized them excessively. So there's another culprit with this disease, glioblastoma, because it's not only this one type of cells, there's 4 different subtypes, and each of them, so it's 4 different types of cells that you can have. So that's called classical, mesenchymal, neuronal, and proneuronal. And they all are slightly different, and they respond differently to drugs, and they express different proteins, and they have different mutations potentially in the genes. So they have gone through a lot of effort and established 12 different cell lines, which represent these 4 different subtypes, and characterize them genetically, so we know exactly which genes are faulty, which proteins are overexpressed, how do they behave, how fast do they divide, do they grow in 2D or 3D? Like, they have already done a lot of the groundwork, so we can take this information and utilize that to assess what are the requirements to respond to this drug that we're going to test better. Like, can they so can these cells, if they respond well, why do they respond well? Or if they don't respond, why don't they respond? Is there something that they all have in common? And so that's the power of this particular tool, that we have these fantastic cell lines, and we're going to do 2D and 3D culture. So normally back in now the gold standard is starting to become more and more a 3D model, but usually we grow cells in 2D. That means on a flat surface on a plastic dish, and that's very much not what's happening in the body. Like, there's no single surface in our body that is plastic, and there's definitely no surface in our body that's completely flat and straight. Like, that just doesn't happen. So 3D is really the way to go, and what that means is you embed you make like a matrix that is something tissue resembling, and you grow the cells inside of it. And tumors often have the tendency, or tumor cells often have the tendency to grow in like a little ball that we call tumor spheroid. And then you can test things like penetration of the drug into the tumor and how fast do these cells die. Because if you have the cells in a single layer, they're all going to die at the exact same time if you hit them with the exact same concentration of drug. You know, it's definitely not exactly what happens in real life. So we're trying to get more and more advanced with our models, and we're potentially thinking even about doing a co-culture model where you introduce other cells, like cells that make vasculature, so that you mimic more the real life situation, and you test the drugs in a setting that is more relevant to what really happens, so that you don't get the nasty surprises of, oh, but this really worked so well in my plastic dish, you know. So you kind of want to, yeah, be a bit more sophisticated there to streamline the process of what happens at the bench and what happens at the bedside with the patients in the end. But that is, yeah, so we're lucky that this tool exists for us to play with, so to say. And then the idea is that we're going to compare, like, how do they respond to the drug in 2D versus 3D? And we're going to use the gold standard temozolomide as a control, and we're going to test that drug versus the new chemotherapy regime, and we're going to test the different drugs singly and in combination, because we have access to all of this also thanks to Dan. He can just take it from the hospital and bring it to us. You know, this is just fantastic. Like the streamline of our collaboration is just, it's just next level. Like, I come from, you know, I've been in different institutes all over the world. I'm originally from Germany, as I said, and back then I was already excited to be working on primary rat cells because they're a bit more like humans, you know. It's like, but now we're like, this is researcher heaven, what we're doing, like translational research at its finest. You know, it's fantastic. And the Illawarra, you know, we might be small, and I moved from Sydney here, and I was worried that my career might be inhibited because there's not so many options compared to in Sydney with all the big institutes and stuff. But you know what, I think it actually did wonders in accelerating my research. And because here's the hospital, and there's Dan, and here's the research institute, and here am I and Marie, and we all know each other, and we talk to each other, we communicate, we find out about each other's research, and then we combine these combine our powers and move forward. You know, I think this would be much more complicated if I would be working in one of the big institutes in Sydney, and it's much more, yeah, I think it's more streamlined here, I must say. So I'm very excited and very happy that life took me here and that I'm now here at Emory, feeling very much at home. So yeah, that has been great. And then one aspect of this project is obviously together with Martin and Carolyn, we're going to do the radiation treatment on the cells, which is, you know, happening after patients have gone home, so after hours. So I said, don't worry, we're not going to interfere with the treatments there, but this is giving us all the tools. So we're treating these cells and these little cell cultures that make tiny little mini tumors, and we're going to treat them exactly as if they were in the brain, and we're going to try and find out as best as possible how can we destroy this tumor in a way that doesn't destroy all the other cells as well. And if we find that FOLFIRI, this new chemotherapy regime, works as well or better than FOLFIRI, we can go straight into phase 2 clinical trials right here at Emory with Dan heading it. So it's fantastic, and it could really actually make a difference in patients' lives in the near future. You know, it's just, yeah, like next level.
So exciting to hear that this kind of research is happening right here on our doorstep in the Illawarra. And it's a great example of how our academic researchers and clinical researchers are coming together and sharing knowledge, and you know, incredible outcomes could arise from this. So thank you so much, Anne-Catherine, for sharing.
No problem. And actually, at the final note of this is that part of this project is only possible because the Illawarra Cancer Carers have donated so much of their time and money to us, raising funds to supply us with well-needed machines and equipment. And it's really hard in Australia to get funding for research, and sometimes we're just short on stuff and funding for people. And the Illawarra Cancer Carers has been instrumental in so many ways. And I just want to mention a couple of things that they have purchased that will be used during this project, and that is the nanostring machine, which can measure expression of DNA and expression of RNA and protein. There's a tiny little machine, but it's also costing $4,000, you know, that we don't just have lying around, that measures the quality of RNA and DNA, and also the quantity of protein, for instance, which is necessary to do downstream analysis on it. Or a machine that's not a very fancy machine, but you just need it, is called a cytospin, which enables you to spin liquids, like from your blood, for instance, straight onto a cover slip, which is like a glass slip, so you can do staining on it and you can look for certain markers and things like that. Because otherwise, it's really hard often to have cells that are not attached to anything growing on a glass cover slip. And this machine also, you know, another $10,000 or whatever it was. I can't exactly remember, but it's just, I just want to say thank you so much. And I'm going to hand over to you now to explain more what you wonderful people are doing.
I think Jeff, you were going to start us off. So Jeff is the president of the Illawarra Cancer Carers. And Jeff, you were going to tell us a little bit, just summarize, you know, I mean, you do a huge amount, but if you could just summarize for our audience what the Illawarra Cancer Carers actually do.
Sure, Kate. Look, thanks firstly, thanks for the invitation to take part in this webinar on World Cancer Day. It's just wonderful to hear from Anne Catron on the research that's going on there, and we've been delighted for many years to be able to support Emory and other university areas in the research that's going on. Just to talk a little bit about the cancer carers, the carers group was formed just about 31 years ago. It was a group of caring volunteers led by a former oncology nurse. Her name was Jenny Dransfield. She actually sort of formed the group, and they began working in the Cancer Care Centre at Wollongong Hospital. And this group got going and got off the ground and helped to build it up to what it is today. It's I think a fairly trusted organisation, with the main mission statement being to provide a better caring environment for cancer patients and their families in this area. We have 230 plus volunteers. No one is paid, and all monies that we raise are spent only in the Illawarra, which I think is very important, and good that people that donate or to us know that fact, that we don't that it's spent within the area. So over all these years, we've raised I think it's about, and I'll check with our treasurer Dave Berry will confirm this. We've raised almost $5 million through donations and various fundraising efforts. I chair a board of 8 volunteers. We meet once a month, and we decide on how to spend the funds that we raise, whether it be on research, on cancer research, or for special equipment. And sometimes we invite the researchers to attend our board meetings, so they can outline their project and explain why they need funds for a particular project. Just to give you an idea of some of the things we do, one of our main activities is assisting to transport cancer patients to and from the Cancer Care Centre for their appointments. We also, we were, before COVID hit us, visiting patients in the cancer ward as well, and offering them support. And of course, the other stuff we do is raising money for as I mentioned, for research and for equipment. Some of our activities, we raise money from various sources. We receive money from funerals, from people that collect donations at funerals. We receive money from bequests. We also run barbecues, Bunnings barbecues, famous barbecues. We run market stalls, and we sell products that are made by a team of wonderful ladies that sew these products that we sell at special craft days and at various functions. We also run a lymphedema clinic, and we also lease out for a very modest amount scooters and wheelchairs, mobility scooters and wheelchairs for people who may need them. So there's quite a lot going on, although COVID has curbed our activities a little bit. Our major function, our major fundraising function is a luncheon at the City South Function Centre every year, where we have something like 300 people come along, and we have a 3-course sit-down lunch, and we have a raffle, and we have some entertainment, and of course we've had to cancel that last year because of COVID, but we are hoping to have it again this year. So there's some of the main things we do. I might just pass over to Dave, because he can tell us a bit more about how we raise our funds and what happens in that regard. Thank you.
Thanks, Jeff. Yeah, I'm the treasurer for the Cancer Carers, and I've been the treasurer now for, I think, 7 years. My function is really to reconcile accounts, money's coming in, and as Jeff mentioned, we then allocate monies out in various directions as approved by our committee or our board. Jeff mentioned the total amount of monies we've raised since our inception, which is heading towards $5 million. What I can tell you is during that time, $1.28 million has been given to the university for research. Now, when we mention research, it's not, as mentioned by Dr. Piper earlier, it's not just simply purchasing equipment, but in many instances, we will fund the employment of research assistants or further researchers. Our latest one, we have agreed to pay the part salary over the next 3 years for a researcher in a private situation. That means we've got to work pretty hard over the next 2 years to keep funds coming in. Majority of our funds do come from donations. This is the Illawarra, and I doubt whether anywhere else in this country we have as many generous benefactors as we have in the Illawarra. We are greatly supported by, for instance, the transport industry in the Illawarra through the convoy. The general clubs in the area, we get club grants. We get donations as little as $20 from grateful patients through to thousands of dollars as private donations. Overall, our donations for this financial year to date add up to $52,000. Now that's within the 7-month period, and we've got to spend that money. We don't always find it difficult to spend it, but by talking to people like Dr. Piper, we do get requests for research as well as equipment at the hospital. It's important that we carry on spreading the word, because we are word of mouth. We're not a government organization. We can't afford to spend money on TV advertising. So it is word of mouth that raises our donations. Anybody got any questions?
Thanks, Dave. Thank you so much for sharing that. And Jeff, thanks for sharing the incredible work that the Illawarra Cancer Carers do. And it's really lovely to see that connection between community donations being managed by yourselves at the Illawarra Cancer Carers, and then really benefiting the community, whether it's transport or supporting cancer patients and their families, or providing funding for research that otherwise quite possibly just wouldn't happen. So it makes such a big difference. Can I ask, why do you think research is such an important part of what you do?
Yeah, I was just going to say that. We would look at research as an advancement in technology. I mean, it was quite interesting listening to Dr. Piper. She stole a lot of my thunder, because I was going to say 10 years ago, 20 years ago, a lot of cancers would have resulted in people passing away. So the ultimate idea with research, in my opinion, is to end up where we don't have cancers, but we know that's going to take a long, long time. But what we have found over the last period is the increase in the comfortability, if that's the right word, for patients with different treatments, and the expected lifespans, as you explained, is longer than it was 10 years ago or 20 years ago. So a lot of people do have a lot of hope. When you talk about 90% recovery rates instead of 50% recovery rates, that makes a great deal of difference. And there are other things. We have one professor who we support, who is our patron, and he is developing, or has developed, a pill which helps the side effects of chemotherapy. So whilst that's not a cure for cancer, it is helping the patients with cancer. So yeah, that's why research is important to us, is to not only look at the end result, but the interim improvement for our cancer patients.
Great, thanks, Dave. And of course, for researchers like Anne-Catherine, as we've already discussed, the funding is so important because it's really difficult to get grant funding. And Anne-Catherine, I wonder, the connections that you have with the community, that helps your research in other ways as well. Is that right? Through things like clinical trials, and you already mentioned that the ease with which you're able to get certain things from the hospital because of your close proximity and because you work so well together. What is the importance for you for grant and community support?
Well, yeah, grants, you know, getting a grant is always a bit of a luck of the draw. To be honest, it's really difficult to get the big grants. So 75% of researchers is funded through the government, which is like the IHMRI or the ARC, which are the big money givers. But you know, you write a very, very complex application that is like 50 pages, you know, and then the success rate is 16%. You know, it's quite devastating, and there is very top-notch research there that just doesn't get funded because it's so it's so cutthroat, really hard to get in, and particularly for someone like me with 3 kids at home, it's really hard to get my publication record up and working part-time. So for me, it's quite important because I might not on paper be as competitive as others in my age bracket, you know, but there's life always comes in between, doesn't it? Like you just have to try and fit it in. And I'm quite the advocate for trying to have a good work-life balance, you know, to have a family and a career and be passionate about your job. Like I couldn't not do my job. I just love it too much, you know. I really want to make a difference, but I also want to be there for my family. So I think there's other ways in how you can get funding, and that is 26% is others, and that's partially the Illawarra Cancer Carers or the Cure Cancer Australia Foundation or the Cancer Council and other smaller organizations that help you out. And I mean, Emory has been absolutely fantastic. I don't know any other institute that is so generous in supporting early and mid-career research. It's like, for me, it's been an absolute blessing working here and being connected in the Illawarra community like this. It's fantastic.
Thanks, Anne-Catherine. And for those of you who don't know, IHMRI, which stands for the Illawarra Health and Medical Research Institute, so we're based on the university campus, but we are a separate entity. We are independent from the university, being a not-for-profit. And our main goal is to facilitate research that's happening that Anne-Catherine has just described. It's about connecting our academic researchers with our clinical staff to really help to expedite translation into patient care and translation of the research into patient care and improve those outcomes. That's our main goal. And we do research into all different types of diseases, not just cancer, also things like diabetes and mental illness, dementia, to name just a few. So yeah, we're really happy to be able to support researchers like Anne-Catherine and have the support from community groups like the Illawarra Cancer Carers. There was one other question that I saw here from Catherine. Anne-Catherine, Catherine wants to know, if your research results in this new treatment, how long would it take to move from clinical trials to approved treatment for patients?
That's a very good question, and that would be an excellent question if Dan could have made it. But it will be much faster, given that we don't have to start at phase 1, where you establish safety and whether or not it's a safe drug to use in people. This is all established. So if this goes well and the phase 2 clinical trial goes well, then it will, I don't know, I don't want to say a number, but it will be faster than it would have been otherwise. I think the phase 2 clinical trials usually take a couple of months, like you want to increase the dose and see, you do it, treat real people, so you come up with a treatment plan, but that is beyond my my scope. I'm sorry.
It's always a difficult question to answer, isn't it? But
it's a very good question. Very
it is.
Yeah, I'm going to ask Dan.
But we all know research takes time, takes a lot of financial resources, and it takes time. But that is a great question. Thank you so much, Catherine. And
given that it's an approved drug and it's used as chemotherapy drug, so it's the best possible way you can start a new treatment, by having a drug that's already used for that same purpose, but just for different tumor, and it's already shown to cross the blood-brain barrier, which is often another complication with things regarding the brain. So yeah, we're really fast tracking this if we're getting good results.
Yeah, that's fantastic. And I've seen there's a comment here in the chat from, I think that's from Senior Professor Marie Ranson, who just wants to make us all aware the Australian state government funding agencies now require researchers in funding applications to show that members of the community are involved in research programs in helping decide priorities and directions. So I know there's a huge amount of consultation that happens with community from researchers. So I just urge anyone in our audience to keep an eye on our website. Part of the things that we do, as Anne-Catherine mentioned earlier, are clinical trials, and we've got a few kicking off either later this month or early March. So I do urge you to have a look at our website. It's IHMRI, IHMRI.org.au. If that is something that you'd like to get involved in, we'd love to hear from you. Oh, is there one more question's popped up? It's a great question, Renee. Thank you. Renee wants to know, Anne-Catherine, could you describe how you got into cancer research? What did you study at uni, and any other steps that you'd like to add?
Oh, that's a great question, actually. And no, it was quite like the faithful sort of discovery. Like, well, I started rare genetic disorders at the Kids Research Institute in Westmead, and we studied a gene that, if it was mutated, it would cause a late onset muscular dystrophy, which was partially due to the cells not surviving damage. So muscle cells, if you walk downhill, for instance, you get eccentric contractions, and your muscle cells have to stretch when they're contracted. So that often causes tiny little microscopic rips in the muscle cells, but they will be repaired, because muscle cells, unlike cancer cells, are cells that don't divide anymore. They're like finally a grown cell, so they need to be repaired. You can't just quickly replace them. So they have this very sophisticated repair mechanism. And one gene called dysferlin, this is what I studied, is involved in patching up the holes by sending little membranous vesicles to the repair site where it's broken. And its closest sister gene, so to speak, is called myofurlin. And myofurlin, we thought, would probably do a similar job to what dysferlin does. So I started investigating myofurlin, and I found out that myofurlin does very different things, and that it actually it's increasing signaling that causes cell growth. And then I did a literature review, and I found that in many cancers, the protein myofurlin, so when the gene expresses the protein, is overexpressed. So more myofurlin in the cell cause the cell to grow faster and survive, have certain survival mechanisms and signaling pathways. So this is how I got interested in cancer, because then I got in contact with a breast cancer biobank and got some specimens, like proper breast cancer tissue. And then I did a Western blot, which is where you can see how much protein is expressed. And I could see that in the breast cancer tissue versus the control tissue from the same person, there's much more myofurlin expressed there. So that's how I started my interest in cancer, by finding that whatever I was looking at was not important for muscle disease, but it was very important for cancer, and that got me hooked. And ever since, I'm very passionate about cancer and cell signaling in particular as well, and cancer cell signaling, because it's very interesting.
Fantastic, thanks for sharing that with us.
No problem.
So I just wonder if we're closely running out of time. Just for our audience members, are there any takeaways that you'd like our audience to walk away with from the webinar today?
Me?
Yeah.
Well, if nothing else, I think there is hope, and the Illawarra can do great things, and we can hold our own on the world stages, at least from my humble experience between Germany and Australia. I think we can be proud of ourselves, which might sometimes be forgotten, because many Emory researchers actually are born and bred here, so they might not know what world-class standard this is and what an amazing institute this is. So if nothing else, have faith, have hope, and we're working on it.
Thanks, Anne-Catherine. Jeff, Dave, is there anything you'd like to add to wrap up the webinar?
Just like to say, it's very exciting, isn't it, to hear about what's going on, and we're just delighted to be able to help out the best way we can, and thanks to the generosity of the community and people in Illawarra, hopefully I'm sure we'll be able to continue to do that.
Yeah.
And if anybody wants to check out what we're about, we've got our website they can go on, and you can also make donations on the website.
Yeah. Illawarra Cancer Carers.org.au.
Correct.
Yeah, well, thank you so much, Jeff and Dave, for all your support, and to Anne-Catherine for coming on today. I'll share any resources that we've spoken about, so including the Illawarra Cancer Carers website link in the follow-up email that I'll send around to everyone that's registered, along with a recording of today's webinar, in case you'd like to share it with some people in your network. But a huge thank you to everybody for making today happen, including our amazing audience. Thank you so much for all your questions. And to Anne-Catherine, Jeff and Dave, again, thank you so much for joining us today and sharing your experiences and telling us more about the Illawarra Cancer Carers and the great work that you do. Your hard work and dedication, all three of you, makes a real difference to patients, cancer patients within our community. So we really do appreciate all of your support. To our audience, if you haven't already, please follow our Facebook page. We have regular posts about successes that we've had, any clinical trials that we have going on, any events that we've got coming up. Fingers crossed, all being well, we're going to have lots of events in 2022. So keep an eye on that, and check out our website, imri.org.au. There's lots of valuable information about the whole range of research that we cover at Emory. If you haven't already subscribed to our newsletter, that can be found at the Emory website, imri.org.au, and that can be found under news. And finally, at the end of this webinar, there will be a short survey that pops up. I think it's only 3 or 4 questions, short questions, just to find out what you, our community and audience, want to hear more about, and that will help us to plan our webinar series for this year. So look out for that follow-up email. Once again, thank you so much to our audience and to our panelists. It's been wonderful to spend this Friday lunchtime with you. Thanks so much. See you next time.
Thank you, Kate. Thank you.
Bye.
Bye.
Bye.